Okay, so recording is in progress and let me get started. So welcome to the kickoff meeting of the AI Codeathon 2.0, everyone. My name is Maulik Shukla. I'm chairing the Codeathon Coordination Committee. With me, I have members from BRC, PDN, and BRC Analytics. So this event is kind of co-hosted, co-organized by the three BRCs and with support from NIAID. And what we will do during this the next 1 hour is I'll walk you through the purpose of the Codeathon, the logistics, some rules or the guidelines for how we are going to conduct ourselves, and then we'll dive deeper into the proposed projects and the project teams. Okay, so it is going to be a lot of information over next hour, and I'll have to kind of cover it rather quickly, and I won't be able to go into too much detail, but we have set up a Slack channel, and I encourage everybody to kind of get activated, join the Slack organization, join your corresponding project teams, and then use it actively to get more information or start the discussion or get to know each other. Okay. So what I'm going to do, I have prepared a set of slides and I'm going to walk you through that. So let me see if I can share my screen here. Okay, you seeing my screen? Yes.
Yeah.
Yep.
Okay, and let me see if I can put this in the presentation board. Okay, are you still seeing it?
Still good.
Okay. Please mute yourself unless you have a question or something, because I'm getting some background noise from maybe one of the participants. So if you can do that, that'd be great. Okay, so welcome to the NIAID BRC AI Codeathon 2.0. It's going to be hosted September 16 to 18 at Argonne National Laboratory, which is at the outskirts of Chicago in the suburb called Lemont. So from the O'Hare International Airport, it's about 30-minute to 45-minute ride. From Chicago City itself, downtown, it's anywhere from 45-minute to 1.5 hours, depending on the time of the day and traffic. So we are looking forward to, we had over 100 applications that were submitted for the Codeathon, and we down selected the participants based on the background, the experience, and the skill set, and you are all the chosen ones. So welcome to the club. So this is the second Codeathon that we are hosting. We hosted one last year, and that was our first Codeathon. We learned a lot of things over there. It was a lot of fun also, and we'll talk about that in a minute. So this is the second in a series, and we are looking forward to having you all coming to Argonne, participating in the Codeathon, and then learning from each other. So the Codeathon, the purpose of is to bring together domain scientists, bioinformaticians, AI and ML researchers, and the software developers together, form these diverse teams, and then leverage each other's expertise, skill set, and complementary skills to address the larger challenges in the domain of bioinformatics, biology, and infectious diseases, to be more specific. And the primary purpose is to develop AI-enabled solution for the infectious disease research. It is hosted or organized by the three BRCs. So we, as I mentioned earlier, so BRC, BRC Analytics, and the Pathogen Data Network. And these three resources provide a rich set of data analysis tools, workflows, and things like that. So the idea here is to use AI and the capabilities that are provided by these resources to solve the larger problems. And focus on the collaboration and building working prototypes. So go within 3-day period from idea to prototype, and in a way so that we can reuse these capabilities or they can be of use to the larger infectious disease community. Again, we are thankful for the all three BRCs for the support and the time that they have put into organizing this event. Argonne National Laboratory, who will be hosting the event and providing the necessary compute resources, as well as access to the large language models and other related infrastructure. And NIAID for actually supporting this, encouraging the idea, and other partnering organizations from NIH. So this is a glimpse of the AI Codeathon 1.0 that was hosted last November, again at Argonne. And we had a large group of people, about 70 people, who worked on 12 different projects with different ideas and challenges. And the outcome of all this project and the Codeathon is recorded and available through the GitHub website. So let me start with what is Codeathon and what to expect from the event. So it is going to be a 3-day intensive collaborative development event focusing on turning promising bioinformatic ideas in the space of infectious diseases into working prototypes. So this is not the place where people will be talking about the things that they have already built. There are not going to be a set of presentations or a set of talks. The primary purpose is to get activated from the very first hour of the first day, and then actively go into this collaborative exercise of building prototypes, training models, evaluating models, and then creating the benchmarks and things like that. The teams are going to be multidisciplinary, combining the expertise from the domain, biological domain, bioinformatics, AI, ML, and data science and software engineering. Again, the emphasis on building, testing, and demonstrating rather than presentations and the traditional training or workshop, right? So there are no formal trainings or workshops or tutorials or anything like that. The project should leverage the existing BRC infrastructure as well as the other existing bioinformatic resources, combine them with the MCP tools, the literature rack systems, the AI, and the large language models in order to accelerate the discovery or the problem solving in the area of infectious diseases. The success here is working together, collaborating, making friendship, having fun, while building working prototypes, uh, evaluated and reproducible proof of concept. But the idea here is not to develop production-ready systems, right? So that this is not an event to work on the deliverables that various projects already have, uh, on their current task list or the project plan and whatnot, but rather bring together like-minded people and work on the problems outside of your current deadlines and current project schedule and structure and things like that. So hopefully people will engage in that spirit and have fun collaborating. So logistics and rules. So before the Codeathon, as of this day, you all should have completed your visitor registration and have got a confirmation from Lisa about the acceptance or approval of your gate pass. There are few people who are still waiting, and we'll be reaching out to you if you haven't received a confirmation yet to make sure that that you have everything in place to come to Argonne. Make sure once you get the visitor registration confirmed, make sure you have made your arrangements for flights, hotel reservations if you are staying at the guest house or nearby hotel. If you have any questions, I think you all know Lisa by now. She's been a big help in organizing this whole event and been communicating with each one of you. So please reach out to her or me and Ron, and we can help you out with if you have any questions related to that. Codeathon itself will be September 16 to 18. We will get started around 8:30 in the morning, and I'll walk you through the agenda in a minute. So plan to arrive a day early if possible, or as early as possible on the day of the Codeathon, right? But we will start sharp at 9. So if your flight comes late, or if you are stuck in traffic, or if it takes you time to come to Argonne, then you'll miss the initial part, the morning session. So I'll be walking through the projects and project teams, and the expectation is that over the next 4 years, each of the project team will will introduce each among themselves, and then get to know each other, and get started on or started organizing using either Slack or creating their own group calls via whatever mechanism that they prefer, and then start gathering information, pointers in terms of what are the data sets they want to work on, with respect to their project, what are the prototypes, if there is existing code or infrastructure that they want to bring in, and defining the more concrete requirement for each of the project, including the problem statement, the use cases, the data sets, architecture, and evaluation plan. So we will talk about completing this project charters. We will need well-defined project charter from each of the project team in order to know what computed resources you will be requiring, and making sure that we have those available before we come to the Codeathon. The purpose of the Codeathon is to build, integrate, evaluate, test, and demonstrate. So that will be the focus for those 3 days. And then we will be making all of that openly accessible via GitHub repository. So everything that will be built during the Codeathon, we want to make sure that the project teams are documenting it and making it accessible to the larger community, which will be the outcome of this Codeathon. And we will also prepare a report, which will be shared with the participants as well as to the external community. The purpose is to build prototype, but we want to build it in a way so that it is reproducible, it is reusable, and hopefully if we do a good job, then some of that can be converted into a more mature and stable product going forward by one of the BRCs or related resources. And we will have a final session on the final day, on the day 3, where each of the team will be giving a 10-15-minute presentation or a working demo of the system that they have built or the outcome of that project and the lessons learned and the next steps. So we have gone through all the applicants and their ideas and the project proposals that they had provided in their application. And we have tried to consolidate them into this somewhat more manageable set of projects by combining the ideas, the stated interest, as well as the priorities and the resources that we have. And while making sure that we have a scope defined in a way so that we can actually deliver on it, or we can build something meaningful within the 3-day period, right? So we don't want projects that are too open-ended or too vaguely defined. But at the same time, we don't want it something so concrete where it's already done by somebody, right? And we are kind of just reusing it. So it's a balance of both. Projects that are aligned with the NIAID mission and priority, because this is sponsored, organized by NIAID and the NIAID BRC. So we want to make sure that the use cases are aligned with the infectious diseases and related research, because that's where we have our expertise. Room for people, about 100 people that will be in the room. This is their primary expertise or the domain that they are working on. So that's going to be our kind of primary application domain that will be, that these projects are kind of revolving around. The projects should reuse and connect or integrate the data and tools from the existing BRCs or the NIAID data ecosystem or the NCBI and other related resources wherever possible, because we also want to use this to kind of understand what are the challenges, or how these capabilities provided by these sources will be used in this new age of where everything is done via AI, large language models, or agentic frameworks, right? And where do we need to, what are the gaps and how we can bridge those gaps? There are multiple projects that are going to be, will have related themes like literature mining, metadata curation, or kind of building the agentic frameworks. And we'll try to co-locate those project teams so that there is some cross-pollination across the projects as well. Rationale for team selection. So we have gone through every applicant, the skill set or the expertise that they had mentioned. And then based on that, we have tried to create this project team, which are roughly about 8 to 10 people in each of the project team. We have tried to select lead and co-lead, but these are mere suggestions. The leads were selected based on the proposed ideas, domain expertise, and their willingness to lead as per their application. But feel free to self-organize within your project team and work however you see fit, right? So none of these things are set in stone. These are mere guidelines to help us get started. Each of the team includes a mix of domain experts, bioinformaticians, AI/ML researchers, software engineers, and they all bring complementary skill to the project. So hopefully you'll have the right mix of people who are able to help you, gather the necessary data, build models, evaluate, test, document, and all those things, and run things at scale on GPUs or HPC infrastructure. Again, each of the team will also have members from one of the BRCs or more specifically BBRC and the Argonne team. So these are the people who are more familiar with the compute resources that are available for the Codeathon, and they can work as chaperone and provide technical support if you need help in porting your code on the compute infrastructure or running things at scale and things like that. The project teams that I'm going to walk through in a few minutes are not set in stone. You are free to reassign yourself if you feel strongly about the project other than what you are already assigned to. But we do need to finalize the project teams in the next week or so that we are not wasting too much time in just shuffling people around, and people can actually get started on the problems. Okay, so these are the 11 project ideas, the proposed projects, the leads or the co-leads, and the corresponding project teams. Again, as you can see, we have done our best to balance it out so that each team has a reasonable number of members. They have a mixed set of expertise, and they have critical mass to basically go from idea to prototype to some viable product. And I'll walk through this in more detail in a couple of minutes. The overall agenda is going to look something like this. So again, as I mentioned earlier, we are not going to have a set of talks except for the very last day, which will be the project presentations at the end of the event. We will get started at 8:30 in the morning. Make sure that you arrive early. By 8 o'clock or so, because you'll have to collect your badge or the gate pass from the visitor center and then find your way to the venue, right? So give yourself enough time. Try to arrive by 8:30 so that you can get to know people, maybe grab a cup of coffee, have some snacks, breakfast, and then we'll get started at 9 o'clock. We'll have a keynote address from the PI of BBRC. He is also the director of the Computing Environment and Life Sciences Division at Argonne and leading the mission genesis for the Department of Energy National Lab. So he has a much broader perspective on the AI and the future of scientific discovery. So we'll try to tap him in to help us get started, maybe with a broader view of the AI and where the field is going. And then we'll get started with the Codeathon overview and the charge. We'll quickly orient you with the compute resources and stuff. And then the primary mode of operation here is that we will have the breakout session. Each project team will have one or two tables where they are co-located, and then they will be working in that teams or groups on their problem and building the prototypes. Okay. In the afternoon, the building where we are, the venue where we are hosting this Codeathon, it sits right next to one of the largest supercomputers in the world called Aurora. So we will have a tour of that supercomputer, and people who are interested, we'll try to have some fun. We'll have icebreaker bingo so that people can get to know each other than the work or what they do. And then we'll try to organize a group dinner in the evening. This is our tradition for all the workshops that we host at Argonne and similar events that we go to a place which is famous, Chicago deep dish pizza plus beer, and we can interact in a more casual environment. Day 2 and day 3 would be very similar to the day 1. We'll primarily working in the breakout sessions and working on our problem states, problems, and the building the prototype. In the afternoon, we'll try to have an organized tour of the rapid prototyping lab. So this is the self-driving lab driven by AI, and it's in the same building. So we'll try to organize a tour of that as well. And then on the last day, in the last couple of hours of the meeting, we will have a report out or the presentation from each of the project teams, and then we'll have the closeout. So we'll try to end by 3 or 3:30 so that people can get their Uber ride to the airport and then can head out. Okay. Let me pause here and see if there are any questions before I go deeper into individual projects and project ideas. Any questions?
Can I
If
ask a
not,
question?
yeah.
So you mentioned that we can reorganize ourselves in terms of projects. And when does that have to happen? Like, is it before the event itself, or can
It
be
needs to
within?
happen in the next 1 week, right? So
Mhm.
I'll show the spreadsheet. Let me do that actually.
Mhm.
I'm glad that you asked that actually. Okay, so here on this slide, there is a link at the end, and I may have it open here. So this is the spreadsheet where there are the proposed teams, and I have added one column which says reassignment request. So if you feel strongly about it, or if your name is not included here by mistake, right, then put your name next to the project that you want to be reassigned to, right? And what
I
we'll
see.
do is I'll go through that and I'll then kind of finalize the assignments, right? But again, as I mentioned earlier, the teams are kind of formed based on the interest and the the projects and the ideas that people had mentioned in their application. So hopefully, I'm hoping that we have done a good job, and there won't be too much shuffling that we have to do. But if you feel strongly, feel free to add your name next to the project that you like to be reassigned to. Okay.
Thank you. That makes sense. Yeah. Thank you.
Yeah. And again, as I mentioned, try to do it if possible this week, latest by the end of next week, because we want these groups to kind of start talking among themselves and share ideas, share pointers, references, and work on the project charter. So if you are deferring the if you are still shuffling the teams, then it will uh, it will be counterproductive. Okay. So, all right. Any other questions before I continue?
Yes, I put a question into the chat, and one of the
Okay.
questions is I don't have any account which was able to access the resources. Are we going to be assigned then?
Right. So we are working on that, assigning the compute resources. So I'm working with the IT team at Argonne National Laboratory. We will be providing access to the large language models. So we have an internal system at Argonne called Argo, which gives access to all the latest Gemini, Cloud, ChatGPT, and GPT and other models. So hopefully, any model that you need access to will be made. Hopefully, we'll have access. You'll be able to access that through Argo. If there are open source models that people want to use, then let us know, and we'll try to make sure that we have them hosted locally so that people can access it. And I'll talk about Project Charter in a minute. Actually, let me talk about it right now. So for each of the project team needs to fill out this project charter. This is the template. Right. So you can pick your, some silly name for your project team if you want, that's fine. Define the leads, the members and their roles. And I want individual project team to fill this out so that it's an exercise so to get get to know each other. Define your project more concretely. What are the goals or objectives that you want to accomplish over the 3-day period? What is your approach, right? So what methods you want to try, what algorithms or approaches that you want to try here, what are the steps in in your proposed solution, and then try to define what data you need, what tools and services you need from one of the BRCs. For example, if you want prepackaged data, then let people know in advance so that maybe either your team or we can work with you to make sure that you have the training data or the test data that you need before the Codeathon. Specify what large language model or the AI models that you want to use during the Codeathon, and what are the compute resources and storage needs that you have, right? Whether you need few GB worth of storage space or whether you need several terabyte, gigabytes or terabytes or petabytes, right? So these things would be helpful, and based on that, we can make sure that we have enough resources available. Now, as we move forward, we will be providing more information about how you can access these resources, okay? The other parts of the charter is what are the expected outcomes, potential impact in terms of the kind of the science or to the field of infectious diseases, and what are the next steps, right? And if you need technical support, then specify those things as well, that what are the things where you need support if you don't have all the skills that you need in your project team, okay? So these project charters also need to be completed by each of the project team by the end of next week, because we will be taking this, we will be consolidating all the requirements from 10 or 11 projects that we have. And based on that, we'll be having a meeting with the IT group to make sure that we have compute resources we need. Okay. Any other questions before I continue? Okay, let me walk through the projects, and then we'll probably leave last 5 minutes or so for questions. So next thing, what I'm going to do is I'm going to give you a very high-level overview of each of the project idea. We have tried to define this as loosely as possible because it is not my job or the job of the organizing committee to tell you exactly kind of what each project should do, right? So all we have tried to do here is to kind of give you some indication of what this project would can be, or what project what problem you can try to solve, what can be the 3-day minimum viable product, or and model or evaluation criteria, and then the recommended team. But our expectation is that each of the project team will take this as a kind of starting point, and then flush it out into more detailed project charter, however they see it fit, right? So use this as a guideline, but not kind of no prescription, right? Feel free to kind of adjust the goals or define your own approaches or evaluation criteria and things like that. Okay, I'll probably walk through first couple of them in a little bit more detail, and then I'll kind of try to keep it at high level for the other projects. So the projects are defined or have a kind of somewhat broader or more generic goal, and within that, what you do is kind of totally up to you. So for example, first project here is the evidence grounded pathogen knowledge extraction. So the idea here is that to automate the knowledge extraction or the curation from literature. This is traditionally has been a kind of it requires manual curation, and it becomes bottleneck as hundreds or thousands of publications come out every year, right? So the idea is to using large language model, RAG, and other similar framework to automate the manual curation process that has been done to, let's say, for example, extract or curate virulence factors, antimicrobial resistant genes, protein-protein interaction, host pathogen interaction, or genotype-phenotype association from literature, right? So the team can identify maybe 1 or 2 organisms that they are interested in, prepare a corpus of maybe a few hundred to few thousand open source publications, build the RAC database, or try different frameworks that that this team member have probably already built or have tried. And then based on that, come up with some criteria, define a couple of data types, and then see how you can extract, let's say, the structured assertions or knowledge from the literature, create your own benchmark evaluation criteria, and then kind of present it at the end. There are different people who have already built their different prototypes. So for example, myself, as well as people who are from BRC here, we have our own RAG infrastructure that we have put together for doing something like this, right? I think there are other people, for example, Vijay Raj, he has a couple of frameworks that he has developed, like Comic and Infectovision and other things that can be leveraged here, as well as Indresh, who has PubMed Miner that he built over the last couple of years, right? So you can also you can try these complementary approaches and then make them off against each other to see where they help, or how they can be complementary to each other, right? So it's okay to try multiple approaches. It's okay to try combination of all these 3 approaches. right? And define your own set of test data, evaluation criteria, and things like that. Okay, so again, it's the job of this team to decide exactly how they want to define the scope of the problem, the approach, as well as what are the test system that they want to use, so that all these approaches can be applied to the same set of data, same set of organism, and kind of evaluation at the end. The second one here is again related to literature mining. So extracting the AMR evidence from literature. So our team here has been working on extracting the antimicrobial susceptibility testing result data from publication for last 10 years or so. And I think we have done this for over 400 publications and manually curated this data. And I think this project can work on automating that entire process and see if we can automatically mine the antimicrobial susceptibility testing results from publication, as well as maybe the curation of the virulence factors and antimicrobial resistant genes to connect genotype with phenotype or something like that, right? So the people here have worked on this kind of problem in the past. For each of the team, I have tried to summarize, I have tried my best to summarize the skills or the expertise or experience from each of the team member. I may not have done that great of a job here, so feel free to kind of update your own description over here if you like. But this is again to for each of the team to kind of at a glance to kind of get a sense of what are the complementary skills each of the team member is bringing to the table. Okay, so this team can identify maybe some subset of paper that can be used as test set, and then work on a framework to automate the process of mining the AST data out of publication. And this is critical. AMR is one of the high priority focus area for NIH and NIAID in general, NIAID and NIH in general. So I think this has a huge value in terms of adding value add to the community. AI-driven metadata curation and harmonization. So this is a common problem. Anybody who's working with the integration of the data sets, or combining data from multiple experiments, multiple genomes for surveillance and things like that, that the metadata is always scattered across bioproject, biosample, SRA metadata, as well as oftentimes publications. And oftentimes it's not where you expect it to be, not using the ontology that it's supposed to use. And then idea here is to use the large language models and AI in general to help automate the curation and harmonization and standardization of the metadata from this variety of sources, right? And then people can also build knowledge graph on top of it to basically aid in enhance the search and retrieval capability for finding related data sets. So the people assigned to this team, they have worked on similar things in the past. And I think each of the BRC as well as NCBI definitely has interest in kind of automating these processes so that we have better metadata available for the experiments, for the genomes, and it helps all the downstream analysis processes. Again, I'm not going to go through this list for each one of them, but feel free to look it over and revise it if you need to. The next one here is sample metadata that survives Galaxy and analysis in Galaxy. So this is a project that is proposed by the BRC Analytics and the Galaxy team. And the idea here is that get the metadata related to the samples that are included in the analysis, be it the genomes or the data from SRA, and then curate it, standardize it, map it to the ontology, and make sure that it is carried forward throughout the analysis process, so that it adds value when you are doing the higher level analysis of the data. So, for example, if you are starting with the SRA data, doing the genome assembly, annotation, and maybe comparative analysis, it would be good if all of that metadata is carried forward, so that when you're doing comparative analysis, all of that is available. Interview driven. So So notice the thing, the first two projects that we had here, they are related to literature mining and knowledge extraction from literature. The next two projects that we had here are related to metadata curation and harmonization. And we'll try to co-locate these projects so that there can be cross-pollination across these project teams as well, and people can learn from each other. The next set of projects that we have here are related to workflow automation and automated workflow execution. So idea in general is to basically take the description of the data or the workflow in a kind of natural language terms from the user, and then automatically deconstruct it into automatically construct a multi-step workflow out of it, map it to the available data and analysis tool, and then execute it. Right. So there is a challenge in kind of user having to find what are the appropriate workflows or tools that map to their problem statement or the analysis that they want to do. What are the necessary parameters, how to run it, how to analyze the result. And the idea for this set of projects that we have here is to automate it using the large language models and AI to the extent possible, using the MCP tools, agentic framework, and things like that. So this is one of these projects, which is basically convert researchers free from analysis request into a validated runnable Galaxy workflow, either by reusing, adapting, or building one, right? So there are already pre-built workflows available. So either you map user request to one of the existing workflows, or you build one from scratch and then execute it. So there are a lot of people from the BRC analytics team. They will be leading this. And then I think we have other people who have shown similar interest are mapped to this team. There is a similar project. This is focused on the BBBRC Copilot or the BBBRC infrastructure. So this is a complementary system similar to, let's say, what we just talked about in the Galaxy system. The idea here is to basically, again, go from allow user to either just upload their data set or describe the analysis they want to do, right? Or point to a publication and then say, repeat this analysis in BBBRC, right? So the idea is to take user input, map it to various multi-step workflow, and then execute it within the BBBRC system. So this team, both of these team will be working on identifying the benchmark or few exemplar workflows based on the publication, or maybe the examples that they have used in the past, and then trying to refine the system or design the system in a way so that it is able to construct this workflow, execute all the steps, verify the output, and then make sure that the whole thing is reproducible and reusable. And one of the key here is to make sure that these AI systems are not hallucinating or making things up that are not necessary or that are not real, right? So a lot of emphasis that needs to go here is basically making sure that it is reliable and reproducible before these systems are made available to the larger scientific community. Federated NIAID data.
Quick question on number 6 is Jason from Los Alamos.
Yeah.
Could this project number 6 actually be treated as sort of a meta project where the agentic workflow would be used to solve all of the other projects agentically, and that would be this Codeathon would itself provide the benchmark. We'd be the John Henry. This would be the steam engine.
Right. So I think the benchmark is one thing that everybody's always looking for. So I think whatever training and test data set or the benchmarks that are used by any of these projects, right, we should try to make sure that they are packaged nicely so that the benchmark themselves can become a reusable thing by other similar projects. And I think there is yes, always opportunity to think more broadly when we are designing this framework to see whether they're applicable beyond a particular resource or framework that being tried here. So the next one here is the federated NIAID data ecosystem. So the last two projects that we saw, they were more targeted towards the BRC analytics or the Galaxy system and the BBBRC. This is somewhat more generic. So the idea here is to apply the same notion or the similar framework, but try to make it work across multiple resources, right? So you can imagine as a user, if you have a question or a problem statement in mind, you really don't care whether the data exists in NCBI or BVBRC, or whether analysis tool exists in Galaxy or BVBRC or somewhere else, right? What you are interested in as a user is to basically find the best possible way to solve the problem. This is how you would be conducting your research, right? If you are a researcher, if you are a PhD student or a postdoc, find all possible ways from where you can get the places from where you can get the data, you can run your analysis, and then at the end combine the result, right? So the idea here is to basically try to come up with a prototype which allow one to combine the data and tools from across multiple resources like BVBRC, BRC Analytics, PDN, NIAID data ecosystem, NCBI, and so on and so forth, and then provide a chat style interactive interface where somebody can pose a question, and then system is capable of answering that question by combining data and tools from all of these resources. There are a lot of infrastructure level challenges here, how do you communicate across multiple resources? How do you maintain identity or the the login or authentication that is required by different system, and so on and so forth, right? So hopefully this team or this project can help identify and address some of those challenges in a way so that it becomes a kind of guideline for other resources or other projects who want to do something similar. Okay, so I think Jason, probably what you were describing earlier, this project is will probably try to address some of those challenges. And we have people from the NIAID data ecosystem, from NCBI, as well as the 3 BRCs, who will be participating in this to take this from idea to some working prototype. The next one here, so now next set of projects, now it's becoming more domain specific, or the problem statement is more grounded in a scientific idea, and then leveraging multiple resources and the MCP tools and the AI or the RAG and other things that we talked about earlier in order to solve a specific scientific problem. So one of this project that is proposed here by the PDN group, the Pathogen Data Network team, is sewage to signal. So developing an AI-assisted wastewater surveillance workflow that basically goes from data to data analysis and then analyzes the result to identify what pathogens are present, what is their prevalence, what are the the prominent mutation, and then also map it or link it with publications and news items and things like that to understand or assess the impact of what is observed in your experiment data. Okay, I'm probably not doing a great job of explaining the the idea in great detail here, but that's the overall concept here. And you can imagine that going from the bioinformatic workflow here, you can combine this with the literature RAG system. You can combine it with maybe other sites that are providing real-time information about the news and things like that, to combine all the information and then inform or add value to the analysis that is done by the bioinformatic workflow. The next one here is structure to function. So the idea here is that traditionally we've been using the sequence similarity-based method to assign the function of the genes and proteins and characterize genes and proteins. Recently you have AlphaFold, OpenFold, ESMFold, all of this new generation of AI-based protein structure prediction tools that have made it possible to make the protein structures available for millions of bacterial and viral proteins. And then the idea is to use these protein structures as well as related advanced AI-based methods to assert the potential function of the poorly characterized or the hypothetical genes and proteins that you have in the bacterial and viral system, right? So the idea here can be that use protein structures to identify to assert whether two proteins interact with each other or not. What are the the interacting or the let's say amino acids that play role in the interaction and things like that. And then once you have the result, of course, you can imagine that the output of this can also be compared or evaluated with the similar output that will be generated by project 1 or 2, mining similar data from the literature, right? So in that sense, these two projects can be complementary to each other. So I think one synergy here can be that maybe this team and project 1 can try to identify or work on the same organism system. For example, let's say influenza A virus or Bundibugyo, which is one of the the recent outbreaks that's happening. And that way, I think we can have a set of results for the same organism system so that it can be compared and kind of evaluated later on. And again, we have tried to put together a team here that have expertise in the protein structures, understand the protein binding, protein-protein interactions, and have worked on similar things in the past. The next one here is hypothesis to omics. And this is where I think I'll be relying more on the teams or the people who have proposed this idea to lead this project. I think the idea here is to translate a biological hypothesis into reproducible search and analysis by using the public transcriptomic, proteomic, metabolomic, microbiome, and other similar data, right? So you can think about this as a multi-agentic system or a co-scientist style framework where basically given a hypothesis, it is trying to find all available lines of evidence from the experiment data as well as from literature in order to assert whether a hypothesis is valid or not, right? And this may require combining the tools and techniques which are we talked about in the previous project, both from the literature, mining related projects, as well as the agentic framework. So again, the key here is that this is unlike maybe some of the other projects we talk about, this is pretty broad in its current form. So I think the team needs to probably work in the first week or so to define exactly how they want to narrow down the scope, make it more concrete so that you can work on it, what kind of data sets you need, so that you can work on it productively during those 3 days. And the last one here is the One Health Conserve Protein Target Prioritizer. I think this is kind of the title that was suggested by someone, or maybe it was just made up using ChatGPT, who knows. But the idea here is that identify the conserved proteins or targets for vaccines and therapeutics for the emerging and re-emerging bacterial and viral systems by combining the genomic surveillance data, immunology evidence, protein structures, host interaction, literature, and so on and so forth, right? So the idea here is to go, come up with a prioritized list of targets for a given pathogen that have the best bet of being converted into can help with the drugs, vaccines, or therapeutics, right? So again, people can identify the organism system, decide what the data sets that they want to work with, what are the resources where those data sets are available, maybe create the compile those data sets in advance, literature as well as literature and other things, and then based on that come up with a approach that combines those multiple lines of evidence in order to help prioritize the targets based on from all available proteins that are in a bacterial or viral system. Okay. So this has a combination of multiple elements, including the protein conservation and evolution and phylogeny and things like that. So this is the 11th project. And in terms of the next steps, let me take just 1 minute here. So the expectation again here, we are about 4 weeks away from the actual Codeathon. The first and most important thing is make sure that you complete your logistics. You have the registration, travel, and lodging and all those things figured out. That's important because you need a place to stay, and we want to make sure that that's taken care of. Activate the project teams. So we have a Slack organization. I included the link to join the organization in my email. So please make sure that you are part of that. So this is the BV AI Codeathon Slack organization. There is an introduction channel. When you join, please introduce yourself, whatever you like to, other people to know about yourself, your expertise, your background. Keep an eye on the general channel because this is where we will be making the announcements and sharing resources or information that apply to all the projects. And then join your project team and then get activated, right? Start discussing ideas, start discussing how you want to develop project charter, share references to the previous work that you have done or other open source software or data packages that you would like people to use and use this space actively, right? Because from this point onward, this is your show. Until now, it was our job to provide initial organization. From this point onward, you are in charge in conducting or running and coordinating your project. So please get activated and use this space actively. If you have any question, you can always reach out to me and others and tag me if you have any questions about the logistics or the Codeathon. Okay. And then, so that is the setting up the collaboration space. Feel free to organize your own meetings for your own project, right? Via Team or Zoom or whatever to get to know each other and discuss ideas. Prepare data and infrastructure. So for many of these projects, you will need to gather data, test data, training data, right? Or start gathering the information on the MCPI or APIs that you want to use, the models you want to use, software and dependencies. So start gathering those things as well. Define your compute needs. Let us know what storage, HPC infrastructure, GPUs, or other resources that you will be needing from us, from the organizer, right? If you think you can run your project out of your laptop with just just using the MCP or API endpoints, I think that's fine. If you need anything extra, please let us know in advance. Try to at least have some homework done before you come to the Codeathon so that you can have productive start, and then define your evaluation and benchmark and things like that. Okay, so these are the set of next steps. Now with that, let me stop here. We have 5 minutes. I can stay on for a little bit longer if needed. So let me open the floor here for if anybody has any questions.
Yeah, quick question. I don't see the project-specific channels in my Slack client yet. Have those been made public yet?
I think so, but if you are not seeing it, then are you in the AI Codeathon Slack? Because we know we have multiple Slack things, right? So
yeah, I'm in the BRC code, and it has the
okay.
In the general,
Curtis,
it has your kickoff meeting comment.
Curtis, click
Okay.
add channels and then
Yeah.
click browse channels.
So not
you should see them.
everybody is added to every channel, right? Because those are supposed to be project specific. You are free to view other channels, and that's totally fine. But its primary purpose is to so you can go here and you can say not create channel, but I think there is
You're
something.
in all of them. I can share my
Yeah.
or the browse channel.
No, I can see them
Right,
when I
so-
browse. Thank you.
Go to browse channel and then you can use this join. Okay.
And then the second question was, you had the template for the documentation and some of the other things, and your screenshots had URLs, but they were too fuzzy to OCR. So is there a place where the links to those documents live?
Okay, so hold on a second. So this is, am I still sharing my screen here? So I think everybody has access to this. So in the presentation, in the last one, there is a link here which says finalize the project charter. And that is linked to, I have created a template for each of the project, right? So this is not the lengthy name that you have, right? But you'll probably be able to easily map the project numbers are the same as what I had in my slide. So go to this, all of these project charters are editable by anyone who has the link. So take the ownership of your project and then start working on that document collaboratively.
Great. Can you drop
Okay.
that top level link into the chat?
Yeah, sure. If I can find the chat here. Okay, please try to access it, and if you have any trouble, let me know. The same folder also has the spreadsheet where if people want to request reassignment to a different project, you'll see that spreadsheet as well as the presentation that I just went over in the same folder.
Great, that link
Okay.
works for me, thanks.
Okay, any other questions?
I did see like there's a GitHub account, but we were not invited to join it as a contributor.
Okay, I'm glad that you are asking this question because even after talking for an hour, I knew I was going to miss something. So we do have a GitHub organization. So let me again share my screen here. This is one of the things I was going to ask. So for many of you, you had provided your GitHub information when you submitted your application. If you haven't done that, go to the same spreadsheet that has these project teams, go to the next worksheet within the same one, and it has your GitHub user, a column for your GitHub username. So if this column is empty next to your name, please find your name and add your GitHub username. So what Danon will be doing, Danon Baker from BRC Analytics, he is the one who is maintaining and managing the GitHub organization for the whole Codeathon. And what he will do is he will take all those username and add all the people to the GitHub organization. And then what he will do is he will make each of the project lead and co-lead owner of the individual project repositories. So once that is done, again, the project individual project leads can take the ownership, add more members, and kind of like modify their project page. Hey, Danon, have we created the landing page for each of the projects? Have you done that?
There's a stub for each one, but we'll flesh those out as we get the full
Okay.
project.
And how do I get to that? I forgot, sorry.
Oh, just go to the, if you go to, well, do you want to see the source or the actual rendered version?
Oh, this is the rendered version.
Yeah.
And then.
Yeah,
Okay.
so each one has a sort of a stub here based on what we had on the slides.
Okay.
Team leads flesh this out as much as you want. Well, this is where like the final project contract will end up or whatever.
Okay. Yeah, so I think once you fill out the charter, either Danon can take them and kind of like automatically kind of populate or modify each of these pages, or you can take ownership of the page and you can do it on your end as well. But the idea here is that I think we had those from the 2025. This is just to give you an idea. Let me pick maybe another one. Yeah, so these are the repositories from the last Codeathon, and you can see here that people have tried to kind of like provide some detail about the project, the plan, and other things, and like enough documentation about the project and team and outcomes and things like that, right? So please walk through maybe some of the previous project as well to get an idea of like how the code and things should be organized here. Okay, what else? So you will all be given access to the GitHub, but please provide your GitHub name in this spreadsheet as soon as possible so that Danon can add you. Other questions? Okay, I'm going to be available on the Slack if anybody has any question, or if you want to hop on a quick Slack huddle or call, let me know and I'll be happy to answer any questions that you may have. But again, as I said, this is not a kind of conference or a set of presentations. The success of the event depends on your active participation, right? So please get activated on Slack and start thinking about your projects.
I do have another question. This is Jason again.
Mm-hmm.
It seems a lot of the projects are sort of at the high level and will end up essentially running in needing to solve the same, often a conserved core set of problems, things that people already will from like named entity recognition. That's always a big one in biology. Predicting molecular binding, just genotype to phenotype prediction in its many aspects. Each project's going to come up with its own tailored solution, or is there going to be some sort of?
So I think there'll be some overlap. I think partly hope here is that, like, there are already shared resources that are provided by the 3 BRCs, right? So BRC Analytics, for example, has a very large kind of like, collection of tools that are available via Galaxy. And I believe the same is available via MCP server, I believe, right? So you should be able to reuse a lot of that. Similarly, BVBRC provides, let's say, a lot of pathogen-related data for bacteria, viruses, as well as analysis services. So if you are working on something that, like, if one of these resources already has the component that you're looking for, then probably, hopefully, you can find a way to reuse it. That is, again, the idea of this Codeathon, right? If you end up developing something new, then again, if it's not available in one of these resources right now, there is a kind of interest to maybe make it, integrate it, and make it more generically accessible going forward. I think there are going to be key infrastructure pieces like name entity recognizer and things like that that may have cross-project value. We don't, given it's only 3 days, I think we'll have to let people improvise and kind of decide what's the best way for them to move forward. But hopefully, these teams we are all going to be sitting in the same room, and hopefully people working on similar things will be co-located, so we can find some way to leverage each other's work in a kind of in a quick manner without having to duplicate the effort. Right. So, yeah, but feel free to, if you have any thoughts along those lines, again, if you are working on one of those components, you can always reach out to other team and say, hey, I have something here that maybe might be useful to you, right?
Right, thank you.
Okay, any other questions? Okay, all right, thank you all for joining. And again, I hope I'll see you all on Slack. And we probably may try to organize another call if needed in a couple of weeks, or just a week before the Codeathon. But if not, then we'll be doing most of the communication via Slack now. All right.
Thank you.
Okay. Thank you for your time.
Thank you.
Bye, everyone.
Thank you so much.
Thanks.
Thank you.